Thank you very much for the introduction. Do you see my screen?
It still hasn't started. It shows you have started the sharing, but we haven't seen the screen.
Right.
Probably need to wait for a few minutes.
Yes, I can say.
Oh,
Yeah,
okay.
I can
Oh,
say.
yeah.
So it works?
Yeah, it works now.
Yeah, great. So thank you very much for the introduction. It's really a great pleasure and a huge opportunity for me to talk today. I would like to thank, of course, the committee members. And today I will talk about the plant microbiota, and the title of my talk is Assembly Rules and Beneficial Functions of Complex Microbial Communities that Colonize Plant Roots. So as you know, in nature, these roots of healthy plants are colonized by these very complex multi-kingdom microbial communities that include bacteria, fungi, and oomycetes that evolved in different kingdoms of life. And in my group, we aim at understanding the fundamental mechanisms that underlie not only the structure, but also the functions of these multi-kingdom microbial communities that colonize plant roots. And as you will see during my talk, we try to combine microbial community profiling data with the use of reference microbial collections and gnodobiotic plant systems in order to dissect how both host microbe, but also microbe-microbe interactions impact microbial community structure and plant health. And I think a major hypothesis in our group is that multi-kingdom microbial assemblages provide greater fitness benefit to the host than individual communities. So over the last 5 years, my group has used what we call the microbiota deconstruction reconstruction approach in order to better understand the assembly rules and beneficial functions of these complex microbial communities. So the first step was back in 2014, a kind of a descriptive step where we understand who is there, what are the microbes that colonize plant roots in nature, followed by a deconstruction step that is involving the establishment of reference culture collections of bacteria, fungi, and oomycetes that can then be used for the design of what we call synthetic microbial communities that are built based on these microbes that we can culture. And then these synthetic microbial communities can then be used for what we call reconstitution experiments in gnodobiotic plant systems that is shown here. For example, where you have a flow pot here, which is a syringe that is cut in the middle and filled with sterile peat and vermiculite. And you can repopulate this sterile matrix with defined bacterial, fungal, and oomycete communities and really test the physiological relevance of the microbiota for plant health. So this strategy has been instrumental in my group because it allowed to move from correlation to causal relationship. And I hope today I can convince you that our work has significantly advanced this plant microbiota research field. So let's start with the first step. What are these microbes and to which extent we can culture them? So the first question we asked back in 2014 is, what are these microbes that colonize Arabidopsis roots in its natural habitats across Europe, and whether there are some core microbes that robustly colonize plant roots irrespective of differences in soil conditions or climates? And to do that, we went to the field across Europe and we harvested Arabidopsis thaliana from Sweden to Spain. And we looked at the composition of the bacteria, fungi, and oomycetes that colonize the roots of these plants. And we were quite surprised to see that although these plants, they really face very different environmental conditions, they often associate with the same small group of highly abundant microorganisms that are listed here. And these core microbiota members are quite diverse for the bacterial microbiota and less diverse for the fungi and for the oomycetes, thus mainly belonging to sodiomycetes for the fungi, Pitialis for the oomycetes. And interestingly, when you look at the names of these filamentous eukaryotes, they have been extensively described as pathogens, especially of Arabidopsis. And I will come back to this during my talk. So the next question we ask is whether these microbes can be cultured and the extent to which we can reconstitute complex microbial communities that resemble naturally occurring root microbiomes. So to make the long story short, my colleagues here and me in my group, we spend a lot of time trying to isolate fungi or mycetes and bacteria from plant roots. And we could show that indeed a large fraction of these root-associated microbes can be cultured. This is what you can see here, where you have the relative abundance of these different microbes in the roots of Arabidopsis grown in the specific soil for bacteria, fungi, and or mycetes. And I have highlighted here in black the microbes for which we have a representative isolate in our culture collections. And you can see that the recovery rate for the bacterial microbiota is roughly 70%, 40% for the fungi, and a bit lower, 30% for the oomycetes. And this has been important because it suggests that it is really possible to reconstitute very complex multi-kingdom microbial communities under strictly controlled laboratory conditions. So in the next few slides, I want to illustrate how this strategy has been instrumental and what we learn from this reconstitution approach here in gonadobiotic plant systems. So the first question we ask is whether these microbes have relevance for plant health, whether these different groups of microbes that include bacteria, fungi, or mycetes, whether they are relevant for plant health, and whether the interaction between these microbes matters for plant survival. So what we did, we recolonized this flow pod system with different community of bacteria, fungi, or mycetes, as well as their combinations, as you can see here. And we looked at the effect on shoot fresh rate of the plant and the survival rate. And we were quite surprised to see that although all these microbes were isolated from roots of healthy plants, they have strikingly different effect on plant performance. And you can see especially these filamentous eukaryotes here, or in combination here, have a very strong detrimental effect on plant health in this reconstitution system, which is fully alleviated by the presence of bacteria, as you can see here. But this means that a major physiological function of the bacterial microbiota is to protect these plants against these filamentous eukaryotes. And in this manuscript, we could further show that the bacterial microbiota can modulate the composition, reduce the diversity, and restrict the growth of these filamentous eukaryotes. And this phenomenon also already occurred in the absence of the host, suggesting that it's really about direct microbe-microbe competition. So the next question we ask is then, what's the relevance of the host innate immunity if the bacterial commensals already provide a first layer of protection? So to test this hypothesis, we recolonized our gonadobiotic systems again with these complex multi-kingdom microbial communities in the context of immunocompromised plants, immunocompromised Arabidopsis that you can see here. And we looked at the relative growth promotion, so the extent to which these microbes can promote Arabidopsis compared to the sterile conditions. And you can see that in the context of wild-type plant, in the context of an intact innate immune system, we see a microbiota-mediated growth promotion, which is impaired here in many of these immunocompromised plants that are indicated here with stars, really suggesting that an intact innate immune system is needed for microbiota-mediated growth promotion in our reconstitution systems. And I want to highlight your attention here to this CYP79B2B3 mutant, which is an Arabidopsis mutant impaired in tryptophan-derived specialized metabolites. And you can see that by mutating these two genes in Arabidopsis, this beneficial microbial community becomes detrimental. And in this manuscript, we could further show that this dysbiotic phenotype that we observed here in this mutant is explained again by the fungal load in the roots of these plants. And this is shown here when you have a very nice correlation between the fungal load in plant roots and the effect on plant performance across these different mutants. So if you take into account what I told you before and this new piece of data, it's very clear that these filamentous eukaryotes are potentially harmful for plants. And they need to be tightly controlled in order to maintain host microbe homeostasis. And they are controlled on one hand by bacterial commensals, and on another hand by tryptophan-derived specialized metabolites. And these two processes act in concert in order to promote host microbe homeostasis and prevent fungal dysbiosis. And if both are missing, the plants are just dead in our reconstitution systems. So the next obvious question we ask is why these fungi are potentially harmful and what are the fungal genetic determinants that drive pathogenicity? So to do that, we performed monoassociation experiment, so recolonization experiment of Arabidopsis using each of these fungi individually, and we looked at the effect on plant performance. So we identify many fungi that have neutral effects, few that have beneficial effects, and again, some of them that are highly detrimental for Arabidopsis. But what I found remarkable is the ones that are detrimental here in monoassociation experiments are the exact same one that we identified across Europe as the core microbiota members of Arabidopsis, so Fusarium, Ilionectria, or Plectosphaera. And then in collaboration with the JGI and Francis Martin, we sequenced the genomes of these fungi and we wanted to associate this phenotypic variation that we see here with genomic signatures in the genomes. And to make the long story short, using two different association methods, we identify a fungal gene family that is called PL1-7, that is important for pectin degradation. So it's a fungal gene that encodes pectinase. And as you know, pectin is the most abundant constituent of Arabidopsis cell walls. So that makes sense that these fungi that are detrimental in monoassociation experiment and that robustly colonize plant roots across Europe, that they are very well equipped in pectin degrading enzymes. But this is, of course, just a correlation. So the next step for us was to take a fungus here, Trichoderma rhizei, that naturally lacks this gene family. And we took a copy of a PL1-7 from a detrimental fungus that we overexpressed in this PL1-7 free genetic background. And what we observe is that this fungus becomes much more efficient at colonizing Arabidopsis roots, and that this increase in colonization aggressiveness was associated with a negative effect on plant performance. Again, suggesting that these fungal pectin-degrading enzymes, they are important for root colonization and for detrimental effect that we observed in monoassociation experiments. So more recently, we became interested in metabolites that are secreted by bacteria and that have inhibitory activity, because we hypothesized that these inhibitory exometabolites are likely important for shaping the root microbiota and also likely for protecting these plants against these filamentous eukaryotes. I will come back to this in a second. But in this part and in this work that I show you here, we ask whether there are bacterial genetic determinants that are important for keeping the competitors away at roots and thereby driving the colonization success of some root microbiota members. So to do that, what we did a very comprehensive screening using this modified Burkholder assay. So basically you have a lone bacterium here, and on top of this lone bacterium, you spot different bacteria. And after a few days, you look at the presence of this so-called halo of inhibition that reflect the fact that this bacterium secretes an exometabolite that inhibits the growth of the lone bacterium. And we screen all possible types of interactions, so 198 times 198 bacteria, resulting in almost 40,000 binary interaction phenotypes. So this huge screening led to the identification of one strain here that we call root41, that is a Pseudomonas brassicacearum, that inhibits almost 25% of the strains in our culture collection. And again, to make the long story short, we identify the genetic determinants in root41 in this bacterium that explain this striking inhibitory activity. So the first genetic determinant is a classical antimicrobial, or is a gene that encodes classical, that is in a biosynthetic gene cluster that is predicted to be involved in the production of an antimicrobial called DAPG, diacylfluoroglucinol. And the other one is predicted to be involved in the production of pyoverdin, which is an iron chelating molecule that can sequester iron and makes it unavailable for competitors. So the next big question that we ask is whether this subtle genetic perturbation that we can apply here in this specific strain root 41, whether this genetic perturbation will have consequence on the community composition and diversity of the root microbiota. So we performed again these reconstitution experiments with synthetic bacterial communities in the context of the root 41 wild type or the exometabolites mutant. And what we observed is indeed that these exometabolites with inhibitory activity, they are important for restricting the diversity at the root interface, but also to shape the community composition. And they are also important to promote root 41 strain competitiveness in the roots, because these exometabolites, they allow the strains to grow better in the root compartment compared to the exometabolites mutants. Sorry, I have something that... Do you still see my screen? Yeah. Sorry.
Yeah, we still see the screen, but are you trying to show a video or something?
No, that's fine. I had a pop-up that was on my screen, so I had to remove it, but that's fine.
Okay.
So one very interesting observation is that these bacterial genetic determinants that are important for bacterial competition, we recently realized that they are also likely relevant in the context of bacterial fungal competition. And what you can see now here in the loan is different fungi from the core microbiota. And these fungi are detrimental again in monoassociation. And you can see that root 41 is also able to inhibit these fungi. And this inhibitory activity fully depends again on DAPG and pyoverdin, really suggesting that those are exactly the same genetic determinant that protect, or that, let's say, inhibit fungal growth and bacterial growth in this system. So the current model is that root 41 is a very strong competitor in the microbiota that can produce at least two inhibitory exometabolite. One is a classical antimicrobial, one is an iron chelating molecule. And although these two inhibitory exometabolites have completely different mode of actions, they co-function together in order to inhibit a broad range of root-associated bacteria and fungi. And by keeping these competitors away, this drives, of course, the niche-free and the colonization success of root 41. We also observe that this mechanism is only activated in the roots, and we think that this is because the iron availability is low at the roots and the amount of photoassimilates is high, which is not observed in the surrounding soil. So we are not only interested in bacterial inhibitory exometabolites, but also about conserved root colonization determinants that are important for bacteria to colonize plant roots. And this is quite tricky because we want to understand these conserved determinants that are activated by multiple strains during root colonization. And recently, my group succeeded in developing what we call a genome-resolved metatranscriptomics that allow us to understand transcriptional reprogramming of individual strains in a microbial community context, and to identify not only strain-specific processes that are activated in roots, but also more conserved mechanisms that are activated here by multiple strains during root colonization. And we further validate that these conserved genetic determinants are indeed important for root colonization in this strain called root 179, which is a rhodonobacter. And you can see that when we inactivate these genetic determinants in this strain, the strain is no longer able to colonize Arabidopsis roots, but remain able to grow in liquid media. So we are quite excited about this because now we can understand conserved genetic determinants in bacteria that are consistently activated by multiple strains during root colonization. So finally, the last part of my talk is about the root-shoot communication and whether root commensals can promote resistance to above-ground stresses. And in my group, we recently demonstrated that the microbiota has a strikingly beneficial effect on the low photosynthetically active radiations, on the low light. So the presence of the microbiota really boosts plant growth compared to the microfluidic control plants, for which you see a growth defect. And this growth increase that we observed in the context of the microbiota is associated with a increased susceptibility to pathogens, suggesting that these plants, they prioritize growth over defense under low light. And we could observe that this phenomenon is completely impaired in a mutant called MYC2-3, in the Arabidopsis mutant. And we observe that this investment into growth is impaired. And instead, here we observe that the plant is prioritizing defense over growth in the context of the microbiota. So this led us to the important discovery that response to light above ground and response to microbes below ground are actually connected along this root-shoot axis, and that this is dependent on the transcriptional factor MYC2, and this is also dependent on the composition of the root microbiota. And basically, under low light, the plants really change the structure of the root microbiota in order to boost microbiota-induced growth over defense responses under low light. So finally, a general model. So I hope I could convince you that our reconstruction or deconstruction reconstruction approach in gnatobiotic system has been important. I hope I could convince you that roots of healthy plants are colonized by multi-kingdom microbial communities, that the filamentous eukaryotes are potentially harmful for plants, and that they are controlled on one hand by the host innate immune system and on the other hand by bacterial commensals. And we believe that once these filamentous eukaryotes are under control, then they provide important beneficial activities to the host. I hope I could also convince you that now we are trying to identify mechanisms and genetic determinants that are important for root colonization on the microbial side, so in the bacteria and in the fungi. We also identified genetic determinants that are important for controlling these fungi on the Arabidopsis side, but also on the bacterial side. And finally, we identified plant genes that are important for balancing microbiota-induced growth over defense responses under low light. So with this, I want really to thank this really bright team of people. I want to thank all the mentors that have been very important for me during my scientific path. I want to thank the funding, Max Planck, ERC, JGI, DFG, and CPLAS. And I want to thank, of course, all collaborators without whom this work would not be possible at all. And thank you very much for your attention.
Thank you. Thank you, Dr. Hackworth. This is a very interesting talk. And now it's time for our Q&A section. Any questions from the committee members?
Yes, hello. I just have some very basic questions. I'm not a microbiologist. I'm not in that field. Sure. I wanted to know, when you talk about root colonization, what does that mean? Does it mean apoplastic colonization, and is it restricted to the ground tissue, or do you also see that inside the stele?
Yeah, well, we mainly focus on bacteria that can propagate in the rhizoplane, so really at the outside of the root. We are trying, of course, to identify endophytic colonizers, but at the moment, it seems that many of the bacteria that we have in the culture collections are very well equipped to colonize the outside of the roots, let's say the rhizoplane and the rhizodermal cells. And only few of them seems to be able to endophytically colonize the roots. So that's a very important distinction. And I think in the work that I presented today, we are mainly interested in the bacteria that can really propagate and reproduce, let's say, at the roots, but not necessarily inside the roots.
Inside. And then you had this interesting collection, and then the recovery was quite differential. You said 2/3 of the bacteria, I think you could culture. What is the general reason for a species not being able to culture? I mean, is that known why you cannot retrieve all of them?
Yeah, well, I think it's about, you know, carbon source or nutritional specificity. So this reminds me, for example, mycorrhizal fungi, you know, that were known to be unculturable for decades. And now it seems that by using different lipid composition, I'm not an expert, but I think now there have been some advance in this field. So I personally believe that most microbes can be cultured if you find the right condition to culture them. It's just about to find the right conditions. But of course, in our work, we mainly focused on 4 different growth media. So that's the maximum we can do for the fungi, for example. And I think for the bacterial microbiota, they focus on 2 growth media. And of course, we primarily capture the fast growing strains. And sometimes we, it's easier to, let's say, capture the fast growing strain than the slow growers. So for sure, there is a bias in our culture collection that we understand, that we know. The big question is whether these microbes would have a big effect or not on the structure and the function of the microbiota. And this, I must say, we don't know. So that's a little weakness, you know, that we cannot culture everything. And it would take a lot of time to adapt growth media for different strains and try to really culture them all.
And then just one little detailed question at the end, if I may.
Sure.
So you showed this this fascinating compensation of growth in low light, right?
Yeah.
Your graph there was a canopy size in square centimeters, I think. Does that translate into biomass, or is? Yeah.
So there is a very good correlation. At least we just looked at correlation in this work, and there was a good correlation between canopy size and shoot biomass. We analyzed both. Yeah. But this is true for Arabidopsis, might not be true for other plant species.
Okay, thank you.
Welcome.
Hi, Stephen.
Yeah, hey, hello. Nice to meet you.
Very exciting talk. It's very exciting. You show that the different microbial interaction among fungi and bacteria, even oomycete. But we also know that the environmental conditions such as pH or nitrogen or phosphate in soil could also hugely impact the microbial. So currently, it is possible to count the different interaction in the microbial interaction. Is some method I can use to quantify the interaction among the microbial and the different?
Well, yeah. First of all, I think it's a very good point. I mean, in the experiments I showed, you know, we mainly focus on normal conditions. You know, we use peat and MS medium, but now we realize that, of course, plants are not, you know, and roots, they are not just this, you know, they interact also with the leaf and with the soil, and there are connections along this root shoot axis that we called. And now we start to realize that it's very important to maybe use our deconstruction reconstruction approach more at the ecosystem level. So you can also, for example, deconstruct a soil and reconstruct it, or you can deconstruct a environmental condition and reconstruct them under strictly controlled laboratory conditions. And we think it's a very good strategy to move from or to try to understand causality in an ecosystem context. And we try not to manipulate nutritional parameters like CO2. You know, we really try to apply perturbations in our systems, perturbations that we can measure and quantify, and we look at how these perturbations affect the composition and the functions of these root-associated microbes. So I think we are now moving in this direction, trying to integrate more findings into a more ecological context where, of course, plants are under stress and they face multiple biotic and abiotic conditions that they must deal with. Yeah.
Okay, thank you. Very exciting.
Okay. Thank you, Dr. Hackworth, for the very interesting talk. And now